Exploring the early history of life. The early history of life was a part of many of my projects as an active scientist, and for me, this is an exciting topic. I've already presented an overview about the history of life on Earth on this channel, as well as a video about a very ancient form of life, the cyanobacteria. Here I want to demonstrate how it is possible at all to find out about these ancient times. As an example, I will present an important topic from the early development of life, the development of our cells. But first, let's talk about methods. How do we know how organisms have evolved? Fossils help for sure, but they are available only partially. The most important approach for finding out about the past is based on the assumption that similarities of organisms imply relationships. By grouping organisms according to the similarities, you automatically obtain the relationships, don't you? Well, as we will see for vertebrates, it's not always that simple. For this approach to work, you have to select the right features for your comparisons. Here are some example vertebrates, and it is certainly not too difficult to group them according to similarities. Let's take, for example, all spindle-shaped animals with flukes. Are these organisms really closely related? These two here are separate from the rest since they have lungs and a completely different skeleton. In the end, internal organs and the skeleton are more important for defining vertebrate groups than external shapes. As a result, seahorses are also fish, although looking very different from the outside. When grouping vertebrates according to their skeleton and their internal organs, we end up with the five groups you all know from school. How are these groups related to each other? Which group has split apart from the main group a long time ago, and which group has split apart only relatively recently? Again, we have to look at the similarities. Similar organisms are closely related to each other with relatively recent common ancestors. Organisms with many differences, on the other hand, have common ancestors only way back in time. In this case, fish certainly show most differences with respect to all other groups. For a start, they don't have lungs, legs, or arms, though their common ancestor with all the other animals must have been alive a very long time ago. The next group with large differences with respect to all other animals here are the amphibia. They do have lungs and limbs, but they still have many features fitting rather to aquatic animals than to terrestrial ones. So this group probably separated as the second group from the vertebrate mainstream. Now, how are reptiles, birds, and mammals related? Birds and reptiles are more similar to each other than to mammals, though they are particularly closely related, sharing a relatively recent common ancestor. Mammals have split off the bird-reptile branch before these two groups began to separate from each other. In summary, we have produced a simple visualization of vertebrate relationships. Fossils can corroborate the branches shown here and, above all, allow dating the branching points in this tree. But how does this work for early life? How can we find out about relationships without any fossils? First, let's try grouping all organisms. Which groups could we define? I think most people will agree with a grouping like this. Plants, animals, and fungi are widely accepted as basic groups, at least for macroscopic organisms. This grouping becomes blurred, however, when looking under a microscope. Therefore, I added microorganisms as a further group in this setting. These microorganisms, however, comprise very different forms of life. They are organisms with very complicated cells containing nuclei and other organelles, this way resembling our own cells, but also organisms of a very simple shape. Since complex cells with nuclei are found in all macroscopic organisms, we can fuse plants, fungi, and animals with respective part of microorganisms into a group called organisms with nuclei. The other group apparently has a very simple cellular construction plan and is called organisms without nuclei, or in former times, simply bacteria. How have these two groups evolved? Here's a very simple model. After a common origin, both groups have separated from each other a very long time ago and evolved separately. Why do we suppose a common origin? Because both groups use the same genetic language, the same language to read and utilize their genes. There is no way this language might have evolved in precisely the same way twice. When the two groups separated, the common genetic language must have been all around already. Apart from this common origin, however, the whole thing seems a bit boring, don't you think? This simple picture radically changed about 50 years ago when genes, DNA molecules, started being used for disentangling genetic relationships. DNA molecules are long chains constructed from four different building blocks A, G, C, and T. They function as genes used by organisms to pass their features to their offspring. In the course of this transfer from generation to generation, mistakes occur and accumulate randomly in the molecules. The number of deviations between two groups refers to the time which has passed since these two groups have separated, since they had their last common ancestor. Take this as the section of such a gene from a shark, a DNA molecule of 50 building blocks. To find out about relationships among fish, we should first determine the composition, the sequence of this DNA molecule in other fish species, like this. Then we have to look for differences between the sequences of building blocks. In our example, the molecules from rays show 2% deviation from the shark molecule, while herring show 3.5%. Therefore, rays would be related more closely to sharks than herrings. Their last common ancestor with sharks would be passed a shorter time ago when compared to the last common ancestors of sharks and herrings. This way, relying on DNA molecules, it is possible to determine relationships for organisms without clear differences in shape, like for many microorganisms. It is even possible to date branching points once you have determined how many deviations are incorporated in DNA molecules over a certain time period. We could determine in our example at which time point sharks and rays or their ancestors and herrings have separated. This method has been applied about 50 years ago for exploring relationships within the large group of organisms with nucleus, formerly known as bacteria. As a result, scientists found out that this group of organisms actually was composed of two groups, which must have separated a very long time ago. One of these groups retained their former name, bacteria. The other group consisted of organisms often thriving under extreme conditions like boiling temperatures, high acid or salt. These organisms were named archaea, and they constitute indeed a very interesting group of life. Unexpectedly, however, the most interesting aspect of this study concerned a group of organisms not really in the focus of the analysis, our own group of nucleus containing organisms. The genes from our group were intermediate between the genes from bacteria and archaea, with sequences rather resembling archaea than bacteria. But even this is not the whole thing. Our cells contain most of their DNA inside the nucleus. Many sequences from this location actually rather resemble archaean than bacterial sequences. But we have other locations with DNA as well, the mitochondria, here labeled in red color. Which organismic group has DNA most closely resembling DNA from these cell organelles? It turned out mitochondrial DNA rather resembled bacterial than archaeal DNA. Even before this discovery, mitochondria had been suggested to be remnants of formerly independent organisms involved in symbiosis with the precursor of our cells and increasingly integrated into these cells in the course of evolution. The comparison of DNA molecules suggested that these two symbiotic partners came from completely different branches of life, and our cells, and basically all living organisms we can see with our naked eyes, are chimeras composed of these two branches. This brings me to the end of this video. I have shown you how scientists nowadays analyze the relationships of organisms by studying their similarities and differences. By including DNA molecules into such comparisons, we can also address relationships of organisms without clear external features. This way, we considerably widen our horizon, allowing a peek even into early evolution of life. An important event from this early evolution was the formation of complex cells with nuclei and other organelles. Such cells constitute the basis of all larger organisms living on the planet today. They have formed by the fusion of representatives of two very distinct branches of life. Life apparently not always has evolved as directly and unilaterally as many see today. The tree of life in parts rather resembles a hedge than a tree, and the formation of mitochondria won't be the only example for linking points in this network, which I will present here.